Hello everyone, my name is Snehlata and I'm a PMRF fellow in the Department of Biological Sciences and Bioengineering at IIT Kanpur. Today we will continue with the next lecture of the chapter Photosynthesis in Higher Plants. So till now we had studied about the basic experiments that showed what are the components that are important for photosynthesis, and then we moved from there to the light reaction because we saw that in photosynthesis there are two types of reactions, light reaction and dark reaction. So in previous class we have almost completed the this light reaction, but and there's some parts are left that we will discuss today, then we'll move to the dark reaction. So in previous class we are studying about the non-cyclic photophosphorylation, and we saw that why it is called non-cyclic. It is called non-cyclic because of its shape. As we can see, the electron that is going from here, it is not coming back, right? So there is non-cyclic movement of the electrons. So it is called non-cyclic because of the non-cyclic flow of electrons, right? Flow of electrons. Now, in last class, we have seen that how these electrons move, that it first moves from this photosystem 2, it receives sunlight and then it excites its electron to electron carrier Pa followed by PQ, cytochrome B6F, PC, and then the first photosystem like PS1. And then from here it gets accepted by FES, and ultimately it reaches to NADP plus, which converts this NADP to NADPH, right? And the enzyme that catalyzes this reaction is called as FNR. This is the enzyme that is ferrodoxy NADPH reductase. So all these things we had covered in the previous class. Now the question is that here what we are seeing, that electron is getting excited and then it is getting transferred to the electron carriers, these electron carriers, right? So these are what electron carriers that comprises of electron transport chain. So they comprise of electron transport chain. But the question is how PS2 maintain its electron? Because if the electron is getting excited and is getting transferred to the electron carriers, then how this PS2 here or P680, it is maintaining its electron numbers, because it is important for it to maintain the electron number. Otherwise, at some point of time, it will be completely depleted of the electron and hence it cannot perform the photosynthesis, right? So from where it is maintaining its electron. So this PS2 maintain its electron via this reaction. So the reaction is, you have water molecule. This water molecule, it's get splitted into H plus, electrons, and O2. Now, if you try to balance this reaction, what you can see that for one molecule of O2, how much molecule of H2 will be required? It will require 2 molecule of H2, right? Then only 1 molecule of O2 will be formed. And by this, the number of hydrogen ions that will be released is 4. And the number of electrons that will be released is also 4. And this electron that is generated here by this water splitting, it is then used by this PS680. Okay, so this electron is being used, and hence the electron transport chain, the electron movement that we are seeing, it's not just one electron, it is 4 electrons. Okay, so this number is 4 electrons. Okay, it is 4. So now because this water is splitting, so there should be something that is splitting this water, right? Otherwise it cannot do it by itself. So what are the complex, or you can say what component is important for this water splitting? So the component that split the water is known as water splitting complex. So it is known as water splitting complex, or this is the one name. The second name is oxygen evolving complex. Oxygen evolving complex. Why? Because in this case, this oxygen molecule is being formed. And the third name of this is photolytic complex. So you can call it either water splitting complex, oxygen evolving complex, or photolytic complex based on the function, right? Because it is splitting the water, it is evolving the oxygen, and it is called photolytic. Why? Because this water splitting is happening in the presence of photo, means light. Okay. Now, so now we understood that how the electron is being maintained by to the PS2. Now, the second thing is, what are the products that are formed? So, products. What are the products that are formed here? So, the first product that we are seeing is what? NADPH, right? So, the first product that is formed is NADPH. Now, the second product that we can clearly see is what? Is this oxygen molecule. So, oxygen is being formed, right? And the third product that is not shown here, but we will see in upcoming slide, is the ATP production. So as this electron moves, they lead to certain changes that leads to ATP synthesis. Okay. So through this non-cyclic photophosphorylation, these are the three products, that is NADPH, oxygen molecule, and ATP synthesis takes place. Now, one more thing. If you remember in the last lecture, I was telling that this complete reaction, where does it take place? So this reaction takes place on the thylakoid membrane, right? So this complete reaction from here to here, everything, it is where? It is in the thylakoid membrane, right? And this water splitting, where this thing happens? So if you remember the PS2, where there is PS2 and where is PS1 located? So the PS2, it is located in the inner thylakoid membrane, right? In the non-exposed part. So this PS2, it is located in the non-exposed part that I showed in the last lecture, right? And this PS1, it is located in the exposed part. So it is located on the outer thylakoid membrane, okay? And this water splitting is associated with what? PS2, right? So where this water splitting will take place? It will take place on the inner side of the thylakoid membrane, right? Or you can say lumen side of the thylakoid membrane. So this water splitting, the location of water splitting will be, you can see it is associated with, first of all, associated with PS2, right? Water splitting complex is associated with PS2. So this complex, it is somehow associated here. So it will be here, right? It will be your water splitting complex that I'm showing by WSC. Okay. And it is where it is occurring? It is occurring on the inner side of thylakoid membrane. Inner side of thylakoid membrane. Or you can also say inner side means lumen side, right? You can also call it as lumen side of thylakoid membrane. Okay. So this is about non-cyclic photophosphorylation, where the important thing is all these electron carriers and then what are the products for and where the water splitting is taking place. Okay. Now, before moving to the Z scheme, let's see what I drew here. This is represented something like this. So as you can see, this pink color, this is your what? Thylakoid membrane. This is your outer membrane, right? This is your outer membrane, and this one is your inner membrane. This is your inner membrane, right? Now, if you look into this figure, so it has not consist of all the electron carriers, okay? Mainly the 3 electron carriers it has, but we have how many electron carriers between PS1 and PS2? We have 4, okay? So, but apart from this, what you can see here is, here you are seeing that PS2 is located here. And this water splitting is associated with PS2. And where it is happening? It is happening in the lumen side or the inner thylakoid membrane, right? So this is how it is. You can visualize that how it happens in the chloroplast, especially in the thylakoid. Okay. Now, let's move to the Z scheme. So what is Z scheme? So Z scheme is nothing, but it is a representation of non-cyclic photophosphorylation. So Z scheme is the representation of non-cyclic photophosphorylation. Okay. Now, what do I mean by representation? So representation means that there is a scale known as redox scale, because here what is happening? It is oxidation reduction, right? But this thing is happening here. So there's scale called redox scale. Okay, redox scale. So this is redox scale is drawn with respect to the electron flow. So if you represent the non-cyclic photophosphorylation on the redox scale, you will see like this structure like this. This is a straight line. 1 second. So it will have a structure that represent the Z. Okay, that will look like Z. So this is your, so here is your PS2. Then this is the electron transport. Then here will be your PS1. Again, the electron will move, and then here will be your NADPH. Finally, NADPH will be formed, right? So if you see the shape, this shape from PS1 to PS2 till here, it is Z shape. And because of this reason, it is known as Z scheme. Okay. So why we call non-cyclic phosphorylation as a Z scheme? Because when we represent what we are doing, when we are representing the electron flow from PS2 to PS1, because when we represent electron flow from PS1 and PS2, from PS1 and PS2 first, so PS2 and PS1, it forms Z shape. It forms Z shape. And because of this reason, it is known as Z scheme. Okay. Now, one important thing that I forgot. So here, this movement from PS2 to PA, this movement is known as uphill movement. Okay. So this movement here, it is known as uphill movement. This is your uphill movement. Why? Because up is we need energy. Okay. So here energy in the form of light is required. But this movement, this from PA till PC till PS 700, this is known as downhill movement. Okay. This is known as downhill movement, because in this case, no energy is required. And again, from PS 700 to FES, this is again uphill. Okay, this is again uphill, because again here you can simply learn it from the directions. Okay, so if it is going upward, it is uphill, and if it is going downward, it is downhill. And then again this will be a what? This will be again downhill, right? Downhill movement. So these are, so in one non-cyclic photophosphorylation, there is 2 uphill and 2 downhill movements. Okay. So this is all about the non-cyclic photophosphorylation. Now let's move to another type of phosphorylation, that is cyclic photophosphorylation. So when we discussed about the photophosphorylation, we saw that there are 2 types. So let's talk about cyclic photophosphorylation. So as the name suggests, what is cyclic photophosphorylation? It is cyclic flow of electron, right? It is the cyclic flow of electrons. Now, when or what in under what condition this flow can happen? So this flow happens when only one type of photosystem is active, and that photosystem is PS1. Okay, so when only PS1 is active, in that case, cyclic phosphorylation happens. So let's draw how cyclic phosphorylation happens. So what I said, that only PS1 is active. So if PS1 is here, or PS1 is also known as PS700, so it is active. So it is active means it is receiving the light, right? And what wavelength of light it will receive? So this wavelength will be what? Around 700, right? So the wavelength will be greater than 680. Because if the wavelength will be less than 680, then PS2 will also be active, right? So this wavelength is greater than 680. So it gets activated and it transfers it to electrons. Now, what do we know, that what is the primary acceptor of PS1? So from here, we know that the primary acceptor of electron of PS1 is FES, right? So it will receive the electron. FES will receive the electron. Now, what will happen next? So, if you look at a non-cyclic phosphorylation, we see that from FES, the electron gets transferred to Fe, that is ferrodoxin, right? But in this case, it does not hold true. So, this FES, instead of transferring its electron to FD or ferrodoxin, it transfers its electron to PQ. And what is PQ? PQ is plastoquinone, right? PQ is here in this reaction. Now, from PQ, we can see that where the electrons will move. So, from PQ, the electron will now move to what? Cytochrome B6F. It will move to cytochrome B6F. And from cytochrome B6F, the next acceptor is plastocyanine, right? And from plastocyanine, it goes to again to PS1. So electron movement in the case of cyclic phosphorylation takes place something like this. Okay, so what is the condition that when PS1 will be active? So condition when PS1 will be active is what? So the first condition is about the wavelength of light. So here the wavelength should be greater than 680 nm. Okay, but at the same time, it should be less than what? Less than or equal to 700 nm, right? Because if it go beyond 700 nm, it will not activate PS700 photosystem 1 as well. Okay, so this is the one condition. Now, what can be the second condition in which only the PS1 can be active? So if you remember the location of the PS1 and PS2, so we saw that if this is your, say, chloroplast, right? And these are your thylakoids, right? And these thylakoids, the stack of these thylakoids is known as grana, and they are connected by this stromal lamellae. And if you remember, the PS1, they resides onto this thylakoid membrane as well as on the stromal lamellae. So if a condition arises in which the light is not reaching to the thylakoid or grana, but it is just reaching to this stromal lamellae, in that case also only PS1 will be active. Because if you remember, the PS2, it resides here on the inner membrane, right? So if the light is not reaching to the grana or thylakoid only, and if it is reaching to the stromal lamellae only, like here, so in that case also only PS1 will be active. So it can also happen when during the stromal lamellae activation. Stromal lamellae photosystem activation, right? So light is just reaching the stromal lamellae. So these are the two conditions in which only PS1 will be active. Now, if we talk about the products, of cyclic phosphorylation, so what things can we see is forming here? So is there any splitting of water? So because here the electron is not getting, it is coming back to the photosystem. So we do not need any extra machinery for the water splitting or for the generation of electrons, right? So here, no water splitting is happening, right? No water splitting is taking place. So no water splitting is taking place, first thing. Now the question is, if there is no water splitting, will there be formation of oxygen? The answer is no, right? Now the second thing is there NADPH production? Again, the answer is no. Okay, so there is no NADPH production because for NADPH, the process should go in this direction, F to FT and then to NADP plus, but this does not happen during the cyclic phosphorylation. So here, no NADPH formation. No NADPH formation. And thirdly, but in this case also, ATP production will take place because for ATP production, whole different machinery is used that we will see in the next slide. So here, ATP production will take place. ATP production takes place. Okay. So in this case, the product that is formed is only ATP. no NADPH, no water splitting, nothing. Okay, so only ATP production is happening. Now, one more thing. If you remember what we said, that what is the use of this electron transport chain when we were studying about the electron transport chain? So we saw that there is certain purpose of this electron transport chain, because nature will not evolve in such a way that it is just transferring the electrons and nothing is happening, right? So at that time, we saw that electron transport chain is important to generate a proton gradient. And that proton gradient in turn leads to the ATP synthesis. So this is the thing that we studied in our previous lecture. Okay. Now we will see why we said those things. But before, let's quickly discuss about what is the difference between the cyclic and non-cyclic photophosphorylation. Okay. So we have studied everything. So let's write in the case of in case of cyclic phosphorylation, which PS photosystem is active. So in the case of cyclic phosphorylation, only PS1 is active. Only PS1 is active. While in the case of non-cyclic phosphorylation, both PS1 and PS2 are active. Okay. Now, this is the first thing. Now, second is, in the case of cyclic phosphorylation, if we talk about the product, so what is the product? We saw only ATP. While in this case, in the case of non-cyclic, the product are both ATP and NADPH. And that together, this ATP and NADPH together are known as assimilatory powers. Assimilatory powers. Okay. Now, in the case of cyclic phosphorylation, photophosphorylation, is there water splitting? No. So here, no water splitting. But no water splitting. But in the case of non-cyclic, water splitting is happening. Yeah. So here, water splitting takes place. Now, in the case of cyclic photophosphorylation, where it can happen? So the cyclic photophosphorylation can happen on both this stromal lamellae and thylakoid membrane. And why? Because PS1 is active and PS1 is present in both on the stromal lamellae as well as on the thylakoid membrane. So the cyclic phosphorylation can occur on both stromal lamellae and granal thylakoid. Right, granal thylakoid. That is thylakoid membrane. But the non-cyclic phosphorylation. So the non-cyclic phosphorylation, it can happen only where both PS1 and PS2 are in close proximity, right? And it is where, so it occurs only on granal thylakoid. Occur only on granal thylakoid. Okay, so it will not happen on the stromal lamellae, because on stromal lamellae only PS1 is present. But for non-cyclic phosphorylation to happen, both PS1 and PS2 should be in close proximity. Now, next, here the cyclic phosphorylation will be active when lambda is greater than 680 nm. But non-cyclic phosphorylation, it happens when lambda is less than 680 nm, because the 680 nm wavelength light will be used to activate PS2, and then the light below 680 can activate both PS1 and PS2. So for non-cyclic phosphorylation, lambda less than 680 nm is sufficient. And then the sixth point is, in this case, in case of cyclic phosphorylation, did we see any electron from the outside? So does cyclic phosphorylation requires electron from outside? The answer is no. So in this case, electron is not required. It is not required from outside. Okay, electron is not required from outside. While in the case of non-cyclic phosphorylation, electron is required. In case of this, electron is required from outside. And from where does it gain its electron? So it gains its electrons from water splitting. So it receives the electron from water splitting, right? Okay. Now, when under what conditions cyclic photophosphorylation happens and under what condition non-cyclic photophosphorylation happens? So the cyclic photophosphorylation, it happens in the anaerobic condition. Okay. So in the anaerobic condition, because here no oxygen is forming, right? And it happens when there is poor carbon dioxide availability. So when carbon dioxide is in very low concentration, in this case, the cyclic phosphorylation happens. So it happens during anaerobic condition and poor carbon dioxide availability. Okay. And in the case of, this is 7th point, right? Okay. So in the case of non-cyclic photophosphorylation, it occurs in aerobic condition. And adequate carbon dioxide availability, adequate CO2, right? So in the presence of adequate CO2 and aerobic condition, this non-cyclic photophosphorylation takes place. So this is the difference. Now, one most important point is that can be asked is, in the case of cyclic photophosphorylation, what is the chain or the electron transfer path? Okay. So here, what we know that in cyclic, the electron from PS1, it goes to FES. So the primary acceptor of electron for PS1 is FES. Okay. Now from FES, where does it go? It goes to plastoquinone, right? It goes to plastoquinone, PQ. Okay. Now, so if someone asks that in case of cyclic photophosphorylation, which is the or what is the primary electron acceptor of FES? So it's what? It's plastoquinone. Okay. Now, from plastoquinone, it goes to cytochrome B6F, that is cytochrome B6F. And from here, it goes to plastocyanine, that is PC. And here again, it come back to PS1. So the primary electron acceptors, we can say if someone asks, what is the primary electron acceptor of cytochrome B6F? The answer will be PC, that is plastocyanine. Okay. Primary electron acceptor means the carrier that accept the electron from the one carrier preceding it, right? So this is the chain of electron transport in the case of cyclic photophosphorylation. Now, in the case of non-cyclic photophosphorylation, what is the chain? So we know that the electron starts moving from PS2, right? And from PS2, it goes to PA. From PA, it goes to again PQ. From PQ, it goes to cytochrome site B6F. From here, plastocyanine. And from plastocyanine, it goes to PS1, right? And from PS1, it goes to FES. From FES, where does it go? It goes to ferredoxin, FT. And from FT, it ultimately reaches to NADP plus, that gets converted into NADPH. So the ultimate electron acceptor in the case of non-cyclic phosphorylation is NADPH, right? And this is the order of electron transport. So this is about the cyclic and non-cyclic photophosphorylation. Now, so till now we have seen that how NADPH is being formed, how water oxygen is being formed, right? Now the next question is how ATP synthesis is taking place. Okay. So before talking about the process of ATP synthesis, let's talk about what do we mean by ATP synthesis. Okay. So ATP synthesis means that suppose you have what? You have a molecule named ADP, adenosine diphosphate. Okay. So your molecule, that is ADP. Now, if you add a phosphate group to this ADP, so we are looking at from chemistry point of view. So you have a molecule named ADP, and if you add phosphate to this ADP, it forms what? ATP. So this is how ATP is being synthesized. right? But what this process is known as, so you can say that ATP synthesis is what? Addition of phosphate to ADP, right? Or in other terms, you can say that ATP synthesis is the phosphorylation. Phosphorylation means addition of phosphate group. Okay. So phosphorylation of ADP. And this is what? ATP synthesis. Okay. Now this ATP synthesis, how how people knew that how ATP synthesis takes place. So the ATP synthesis, over the period of time, different scientists came with different model or theory to prove that ATP synthesis takes place via this way, via that way. But the most accepted theory is known as chemiosmotic theory. So the most accepted theory of ATP synthesis is chemiosmotic theory. Now, what is chemiosmotic theory? Chemiosmotic theory, this is given by, this theory is given by Peter Mitchell. Okay. Given by Peter Mitchell. Okay. And what this theory explains? So the chemiosmotic theory, it explains the process of ATP synthesis in not only chloroplast. Okay. So it explains the process of ATP synthesis in chloroplast because we are studying about the photosynthesis. So it's quite obvious that now we are talking about the chloroplast. But along with this, the same theory also explains the process of ATP synthesis in mitochondria. Okay. So we know that in our body or animal cells, we have an organelle named mitochondria, and that is known as powerhouse of the cell because it provides the energy. So from where and or by what method this energy is synthesized in the mitochondria, this is also explained by the chemiosmotic theory. Okay. So this chloroplast and mitochondria, in both the ATP synthesis take place, and it is done by chemiosmotic. It is proved by chemiosmotic theory. Okay. Now, what are the requirements of ATP synthesis? So, in the case of ATP synthesis, when we are studying about photophosphorylation, we said that proton gradient is required, right? So the first requirement is proton gradients. Now the question is, what is proton gradient? So the proton gradient is nothing but the difference in the concentration. The term gradient means difference. Okay. So gradient means difference. So proton gradient is the difference in the concentration of protons, right? So difference in the concentration of protons, that is H plus ions, right? Now, what difference we are talking about? Where this difference should be? So it is a difference in the concentration of proton across. If we talk about the chloroplast specifically here, so it is difference in the concentration of proton across thylakoid membrane. Across thylakoid membrane. Okay. Now, what does this mean? It means that if you remember, this is your structure. This is your chloroplast. In this is a what? Thylakoid. It is double membrane structure, thylakoid. The chloroplast is also double membrane structure. And this region is known as this region is known as what? Stroma. This is known as stroma. Okay. So the proton gradient that we are talking about is a difference in the H plus ion in the stroma, and in the lumen. Okay, because it is transferring, the hydrogen ion is transferring like this. Okay, so it is a difference in the concentration of hydrogen ion across thylakoid membrane. So this is a thylakoid membrane, right? This is a thylakoid membrane. So, and it is a difference of concentration across thylakoid membrane. And when it is created, so it happens when the hydrogen ion concentration or proton concentration is high. Where? It is high in the lumen. Okay, by lumen I mean this. inside this thylakoid, right? And it is low, hydrogen concentration is low in where? Lower, it should be low in stroma. So this is the proton gradient that we need for the ATP synthesis. Okay. Okay. So this is one proton gradient. Now, second important thing or second requirement of ATP synthesis is, of course, the intact membrane. Because this proton gradient, it is creating across the membrane, right? Across the membrane. So the second requirement is intact membrane. Okay. Intact membrane. Because if there will be no membrane, so there will be no proton gradient, right? And then the third requirement is a special particle known as F0 F1 particle. About this, we will study in the upcoming slide. But for now, remember, so that F0 F1 particle is required for the ATP synthesis. Okay. Now, so these are the requirements. Now, what actually happens during the ATP synthesis? So if someone asks in one line what happens, so in one line you can say that during ATP synthesis, the proton, that is this proton gradient that is created, right? That is increased concentration of proton in the lumen and decreased concentration in the stroma. So from this lumen or from high concentration, this F0 F1 particle, it starts pumping the proton outside or in the stroma. Okay. So what happens is here, this is your thylakoid membrane. Here is a lot of H plus ions. So during the proton synthesis, and this is your stroma, right? This chloroplast, this is your stroma. So the proton starts pumping from lumen to stroma. Okay. So during ATP synthesis, protons move from lumen to stroma. Now, one can ask, like, how is it possible? First, the proton gradient is being created by moving the hydrogen across the thylakoid membrane. And again, the process is getting reversed. But during this process, how ATP synthesis is happening. So now let's see how ATP synthesis happens. Okay. So till now we have talked about the requirement. Now we'll see the actual process by which it happens. So before moving to that, there are various methods. Like there are three methods by which ATP synthesis can take place. So there are three methods by which ATP synthesis can happen. The first one is known as substrate level phosphorylation. The first is called as substrate level phosphorylation. Okay. So for the substrate level phosphorylation, you do not need any this proton gradient. It's simply you phosphorylate the ADP in under any condition. So here is your ADP. Now you add the phosphate and it will simply gets converted into ATP. The second method is known as photophosphorylation that we are studying. Photophosphorylation. And this photophosphorylation, it occurs where? It occurs in chloroplast. It occurs in chloroplast. And the third is simple phosphorylation that occurs in mitochondria. So again, the third is phosphorylation, and that occurs in mitochondria. Okay, and both of these are explained by chemiosmotic theory, right? Explained by chemiosmotic theory. Okay. Now, so this is the method for ATP synthesis. Now, let's talk about how the photophosphorylation or ATP synthesis in chloroplast takes place. So, in simple terms, how it takes place? So, it takes place, I'm talking about chloroplast. And even in mitochondria also. Okay, so let's write it like this. So, ATP synthesis in chloroplast and mitochondria, both. Okay, one second. So, ATP synthesis in chloroplast and mitochondria, in chloroplast and mitochondria, how does it take place? It takes place via occur by F0 F1 particles. Okay, so this F0 F1 particles, these are present in both chloroplast and mitochondria, and by this the ATP synthesis occur. Okay, now let's talk about what this F0 F1 particle is. So F0 is what? So this is your chloroplast. Okay, so this is your chloroplast. Now in the chloroplast, we know this is double membrane. So this is the chloroplast is double membrane. Now in the chloroplast, what do we have? This F0 F1 particle, where it is located. So it is located something like this. Something like this. So this is what? This is your F0 and this large particle is F1. And these are attached. So this is your F0 F1 particle. And it is located in the membrane of the chloroplast. This is not thylakoid, this is chloroplast. Sorry. This is thylakoid. So it is located in the membrane of thylakoid. Okay, sorry. So this is located in the membrane of thylakoid. Okay. One second. Yeah. So it's thylakoid. Now, let's see what is F0 and what is F1. So these two are joined together, but they have two different roles. So if we talk about the F0 particles. So F0 particles, these act as channel. Okay, they act as channel. Okay. Now, what is the role of channel? So channel basically what it does, it allows the movement of something across the membrane through it, right? So if it is channel, something will move across it, right? So the F0 particle acts as a channel, and what it does, so it pumps what it is doing. If it is a channel, it simply pumps. Here we know that we have proton gradient, because of which in the lumen we have high concentration of H plus, right? So it will simply pump this H plus ion. Okay. So it pumps H plus. Okay. And this, if we look at the structure, this F0 is located inside. And because of this, it is known as intrinsic protein. Based on this location, it is present inside, and hence it is an intrinsic protein. Okay. So this is about the F0. Its function is to pump H plus ions. Okay. So it is pumping the H plus ions. Now the second particle is F1 particle. So the main role in the ATP synthesis is of F1 particle. So what does F1 particle do? So F1 particle is basically so by the way, this F0 F1 particle means these are protein. Okay, by particle here it means protein. Okay. So this F1 particle, it is also a protein, and it is a catalyst protein. It is a catalyst protein. Now, what kind of proteins, or what is the name of the protein that acts as a catalyst? We know those proteins are known as enzymes. So simply F1 particle, it comprises of enzymes. It is an enzyme. Okay. Now, if we are talking about the enzyme, what enzyme is the F0 particle? Sorry, F1 particle. So F1 particle is known as ATP synthase. So this particle is known as ATP synthase. Okay. So this is ATP synthase, and as the name suggests, it means it is responsible for ATP synthesis. Okay. And if you look at the location, so this protein is located where? It is located outside or external. So it is known as extrinsic protein, or we can say peripheral protein. Okay. Now, how does ATP synthesis happens? So it's simple. You have a proton gradient created. This is your thylakoid. This is your lumen side, right? And here is your stroma. This is your stroma. Now, what happens? This F naught starts pumping or it starts transporting the hydrogen ion from the lumen to the stroma side. Okay, so it starts pumping the hydrogen ion from lumen to stroma. And as it starts pumping, what it happens when this hydrogen ion it reaches to this F1 particle. So suppose it reaches here, one of the proton. It causes a conformational change in the F1. Okay, so this F1 particle undergo conformational change. Undergo conformational change due to proton, you can say proton or hydrogen ion. And once it get under conformational change, it rotates. And when it rotates, what it does, it converts ADP and PI to ATP. Okay. So it undergo conformational change. So simply what is happening? The proton gradient is there. This F0 particle, it is acting as a channel and it is transferring the hydrogen ion from the lumen to the stroma. So if it will start transferring, it will move the hydrogen ion like this from F0 to F1. And as soon as it reaches the F1 undergo conformational change and it rotates itself, and which in turn help in the synthesis of ATP with the help of ADP and PI. Okay, so this is the process. Now, one important thing that if someone asks that what type of transport is this proton ion, protons. So this F0 particle, it is acting as a channel. And does it require any energy? No, because here we are seeing the energy is being formed. It is not getting consumed. So this pumping of hydrogen ion, this is a passive transport. It is a passive transport. And if someone specifically asks about what kind of passive transport it is, so the passive transport it is because here the hydrogen ion is not simply moving across the membrane. It requires a channel. Hence, this passive transport will be what? It will be facilitated diffusion. It will be facilitated diffusion because something is facilitating the diffusion or the movement of hydrogen ions. Okay. So this is all about how ATP synthesis takes place. Now, okay. So this is a simple process of ATP synthesis. Now we were talking about this thing, proton gradient. We said that proton gradient is important, why it is important. But the question is how this proton gradient is generated. Okay. So first of all, let's write in one line the importance of the proton gradient. So what happens in the proton gradient? What happens due to proton gradient? The proton gradient once generated, it leads to the movement of proton through the channel, right? So the proton moves through channel. And what channel I'm talking about? I'm talking about F0 here, right? Through F0 channel. And this movement leads to what? This leads to the change in conformation of F1. Change in the conformation of, it leads to a change in the conformation of F1, right? F1 particle, or it is what? It is your ATP synthase. ATP synthase. And this, in turn, it does what? It does leads to the ATP synthesis. It leads to the ATP synthesis. So this is the importance of proton gradient because if proton gradient will not be there, so the movement will not take place, right? If on either side of the membrane, there is equal concentration of hydrogen ion or proton, so there will be no movement, right? The movement can only take place when, or in most of the cases, the movement takes place when there is uneven or unequal concentration across the membrane. So because of this reason, proton gradient is required. But the question is, how this proton gradient is generated at the first place? So the question is how the proton gradient is generated. So we know the answer. If we go back to the diagrams that we drew, what we are seeing here is First of all, the entire reaction, if you see in this diagram here, this is your what? This is your lumen, right? This is your lumen. And this side is what? This is your stroma. Okay. Now, if you see this NADPH production is taking place where? This is taking place in the stroma. And it occurs by the consumption of the protons, the hydrogen ions, right? So here in the stroma itself, the hydrogen ion is getting consumed. First thing. So this leads to the difference between the proton concentration in the stroma and in the lumen. This is the first case, right? Now, second case, if you remember, we talked about one electron carrier known as plastoquinone. So this plastoquinone, it not only transfer or carries the electron, it also acts as a pump. So this plastoquinone, it acts as a pump, right? And this pump, what it does, it pumps the hydrogen ion from the stroma to the lumen. So it moves this hydrogen ion from stroma to the lumen. So this is your second way by which the hydrogen ion is getting, or the proton concentration is varying across the membrane. And the third is what? And the third thing that you are seeing is the oxidation of water, right? Here, if you look at the oxidation of water or the lysis of water, what happens? It leads to the formation of oxygen and hydrogen ions, right? It leads to the formation of oxygen and hydrogen ion, and this reaction itself is happening in the lumen. So if you compile all these three, you can see that all this process is leading to the increase in the concentration of hydrogen ion in the lumen and decrease in the concentration of hydrogen ion in the stroma. Okay, so this is how this proton gradient is being created. So let's write. So the first one is what? The first one is use of proton or hydrogen ion by FNR. We know what is FNR. It's pyridoxine, NADPH reductase, right? This is the enzyme that converts NADP to NADPH, right? So use of hydrogen ion by pyridoxine NADPH reductase to generate NADPH, right? So this thing, because this thing is happening where? It is happening in the stroma. So if it happens in the stroma, the hydrogen ion or the proton there is getting consumed, and it leads to the decrease in the concentration of proton in the stroma. Now the second event that we saw is what? Was pump. The plastoquinone that was acting as a pump. So pumping of hydrogen from stroma to lumen by plastoquinone. So it will cause what? It will increase the concentration of hydrogen ion in the stroma and decrease it in the stroma and increase it in the lumen. And the third one is what? Third one is splitting of water in lumen. Splitting of water in lumen site, right? So although this complex that splits the water, it is located on the inner membrane of the thylakoid. But once the water will split, definitely its product will be in the lumen, right? So this splitting of water, it leads to what? Production of H plus ions. And this H plus ions goes in the lumen, right? And by this three way, it causes the generation of proton gradient, that is increase in the concentration of proton in the lumen and decrease in the stroma. And further, the ATP synthesis takes place. Okay, so this is about the light reaction. Okay, we have studied about non-cyclic photophosphorylation. We studied about cyclic photophosphorylation, and finally the ATP synthesis. Now, reaction. Let's solve few questions. So the first question is, which of the metal ion is the constituent of chlorophyll? So if you remember, when we were studying about the structure of chlorophyll, we saw that there is one metal ion, that is what? Mg, that is attached to the 4 porphyrin ring, right? So it has a structure something like this, and then it has a long tail. Okay, so the metal ion that is present in the chlorophyll is magnesium. Sorry. Okay. Now the second question is, which pigment act directly to convert light energy to chemical energy? So, which of the pigment here, they directly convert the light energy into the chemical energy? So if you remember, we talked about two things, a reaction center and the antenna. Antenna were the accessory pigments and the reaction center consists of the chief pigment. And the chief pigment was what? Chlorophyll A, right? Because these antenna, they cannot convert the light energy directly into chemical energy. They simply move or transfer energy to neighboring pigment, and then ultimately to chief pigment, and this chief pigment, it does the conversion. So it converts light energy to chemical energy. Okay, and the chief pigment is what? Chlorophyll A. So the chlorophyll A has the ability to convert light energy to chemical energy. Now the third question is, splitting of waters is associated with photosystem 1, lumen, both photosystem 1 and 2, inner surface of thylakoid membrane. So this by splitting of water, it means water splitting complex. It is talking about the complex. So is it associated with photosystem 1? No, because the photosystem 1 does not have to do anything with the water splitting. Is it associated with the lumen? The answer is no, because although after splitting, its constituents are getting into the lumen, but the water splitting complex, it does not reside in the lumen. So the answer is not lumen. Then third is both photosystem 1 and 2. It is also incorrect. And the fourth one is inner surface of thylakoid membrane. So this is what we understood. This is what we have seen through this diagram, right? Here the water splitting is happening on the inner side of the thylakoid membrane. Okay. So the correct answer will be inner side of the thylakoid membrane. Now the next question is, the energy required for ATP synthesis in PS2 comes from, so we have just seen this. I'm not going to explain. So it comes from proton gradient. The next question is, during light reaction in photosynthesis, the following are formed. So what are formed during light reaction? Now we have completed it. Let's see. So first is ATP and sugar. This is incorrect because no sugar is formed. Hydrogen, oxygen, and sugar. Incorrect. ATP, hydrogen, and oxygen. And the fourth option is ATP, hydrogen, and oxygen donor. No, the correct answer will be, it should be hydrogen ions. Okay. So the correct answer is ATP, hydrogen ions, and oxygen. Oxygen donor is not being formed. Oxygen is formed, right? So the fourth is also incorrect. The correct answer is C. And the last question is, the dark reaction in photosynthesis is called so because. Now we'll start the dark reaction, but we have seen, we had a class about the introduction of dark reaction. So why dark reaction is called so? The first option is because it can occur in dark also. The second option is it does not directly depend on the light energy. The third is it cannot occur during the daytime. And it occurs most rapidly at night. So the correct answer here is B. Because the dark reaction, the light reaction is called light reaction because it requires light energy directly. But the dark reaction, it does not depend directly on the light energy, but depends on product formed during light reaction formed during light reaction. Okay, so this is why this is known as dark reaction, because it does not directly depend on the light. Now in the next lecture, we'll start the dark reaction, and then we will see that how the products that are formed during the light reaction are being utilized in the dark reaction. Okay, so that's all for today. Thank you.